All right, ladies and gentlemen, today we're going to do some short notes on osmosis. So I'm going to have you take notes in your notebook, and we will likely check them in the next class, so just make sure that you do that. So first, we're starting off with a basic definition. So osmosis, simply stated, is the diffusion of water. And though this seems like a simple idea, a lot of students get confused between osmosis and diffusion. Okay, so this time we're looking at the movement of water like we saw in our lab today. So we need a little bit more information just to explain. So in situations, so this is what we're talking about, in situations where the solute or the little particles cannot diffuse through that selectively permeable membrane, so they can't pass through the membrane, if the particles themselves or the solutes cannot pass through the membrane, then the water will move from the area of low solute concentration, okay, so where there's very few particles, to the area where there's a high solute concentration, so where there's lots of particles. So in our lab, we saw that the water was moving out of the cell to the area outside of the cell where there was a lot of dissolved salt, and that was impacting the movement of that water. So then we have a little example. So we're just saying here in this example, okay, let's say, I'm going to get rid of this. Okay, perfect. I'm not sure if you can see that, but I can see there's something on the screen blocking the view a little bit. Here we're looking at a situation in a lab where we're saying we have this specialized container and we have a selectively permeable membrane down the center. So this would be kind of like our cell membrane. We have a concentration of solutes, and I did not count them out. I'm just trying to show there's a higher concentration. Let me grab something. So we have a higher concentration of solutes on this side, so on the left side initially, and on the right side we have a lower concentration of solutes. Okay, so another way that we could say that same information, trying to switch colors, I like to have a variety of colors here, is that this solution on this side is more concentrated. Okay, all right, the solution is more concentrated, and this solution is more dilute. Oops, make that an E. Is more dilute. So that means there's just fewer particles, okay, on this side than there are on this side. Okay, so we're saying this is our initial setup, like for a lab. Okay, so we set it up like this, and we're waiting to see what happens. Okay, so we're anticipating then, just like we saw today, if this would be like the interior of our cell and this was the exterior of our cell, just comparing the two environments, where this one has a lot more, solutes dissolved in it that can't seem to diffuse through the membrane. Okay, and this has a few of those particles in it, but the particles themselves can't diffuse through the membrane. So we're going to see the movement instead of the water. So I'll have you make a prediction, like which way do you believe the water will move? Okay. So let's say we let this sit and then we come back to our specialized solution here. All right, I accidentally drew my particles a little bit more on one side, but that's okay. All right. We would expect to see then that the water actually had moved from the right side into the left side. So I'm going to draw up here that I'm actually expecting, I'll switch as many colors as I can here, that we're going to anticipate here that water will move from this side to this side, okay? And that's what we'd see. So our question off to the side, I'll zoom out again, simply says, well, when will the water stop moving in this net direction? So when will it stop moving, toward the left in order to dilute this this solution? Okay, so really we're trying to think about what's that point. So what's that point of balance called? And if you remember, the point of balance is, okay, when it hits equilibrium. Okay, so when we have a balanced concentration on the left and the right side, then we'd expect that the water will stop moving in that overall direction. Now, water molecules will still pass back and forth, okay? So when we're in equilibrium, it's not that things won't move at all, but we might see, okay, a little bit of water is moving that way, and a little bit of water, okay, about the same amount is moving in the opposite direction. So now it's totally balanced, and there's not going to be any net movement. So we don't see all of the water moving in the same direction, okay? So it's well balanced at this point in time. So then we're doing a little reminder here. So what factors would prevent the solute from moving? So remember the solute, those were the little particles that we had. Okay, I'll just draw a few around there. So solutes are little particles. So what's preventing the solutes from moving? So we talked about this within our last SDQ. So specifically, one of the main factors was the size, okay? So if the size, it's too big. So this means the solute is too big to pass through the membrane. So that would be one. So it's too big to pass through the membrane. Oh, I need to zoom out just a little bit more. So it's too big to pass through the membrane. The other one are these other characteristics. For example, okay, some people said, okay, that polar molecules, because on the POGL it said that the things that could pass through were small non-polar molecules. So they said in the POGL polar molecules. So this is talking about polar, or if they are charged, okay, if they are charged particles, they cannot pass easily through the membrane. Oops, sorry, Rosie's barking, but so the charged particles cannot move through the membrane. And if you remember, we used salt. Okay, so I'm just going to do a little chemistry reminder here. So we used some salt in our lab today. So salt is NaCl. Okay, and this on its own is not charged, but actually, okay, sorry, Rosie's getting a little crazy. When you dissolve salt in water, I'm going to draw another little glass. Okay, so this is going to be a glass of water. Okay, here we go. It's my glass of water. When you dissolve the salt in the water, you're going to get the salt to dissociate, and it'll give you sodium ions, okay, and chloride ions. So these are charged particles. So salt is actually strongly associated with these charged particles, even though if we look at the NaCl itself, okay, we wouldn't assume that it's charged, but it's an ionic compound and will dissociate in water, just leaving us with these charged particles. So all I need you to know is salt means charged particles. That's it, okay? So we're going to move on from there and take a look at some different types of environments. So looking at cells in different environments. So let's start here. So there's actually 3 different types of environments that we could see. They have these kind of crazy names. Right, we're going to look at plant and animal cells in 3 different environments. So I'm just going to have you draw out like a little data table or a little table, and you can put the cells in there. So I tried to draw some plant cells and then some animal cells. So we looked at this hypertonic, isotonic, and hypotonic environment. So I'm just going to underline parts of the word. So hyper, iso, hypo, okay? So first off, we're going to draw some particles again. So I'm going to draw my little solute particles again as just these little dots. So we can just get an idea of how many there are in each area. So we're going to say that this is a cell in an environment. This should look fairly familiar. So this looks like the situation within our onion cell. What I added to the drawing was the nucleus, just because cells have a nucleus. And I added this here. So this is the plant storage space. So I added the vacuole and you'll see that the vacuole is actually still inside. So this is my cell wall, and this is my cell membrane. Still inside that main part of the cell. So remember, the components of the cell are contained within the cell membrane. So now we're looking at this hypertonic environment, hypertonic. So if you think about the word hyper, like if you say that a person is hyperactive, it really means they've got like a lot of energy. So when we're looking at hypertonic, so tonic, you should be thinking about the environment or like the solution it's in, or if you drink like tonic water, okay? So that's the liquid that we have this cell submerged in. So this means that there's a lot of particles. So I'm just going to put lots and lots of particles. Okay, just lots of dots here within my environment. And there's still some materials dissolved within the cell. But what we're going to see then is this net movement of water. Okay. So I add this water, okay, moving out of our cell and into the environment. Now next to it we see that we have our isotonic environment. So iso is like equal. So in this case, we have a low concentration of particles, okay, on the outside, or lower maybe, and a lower concentration of particles on the inside. And, you know, we have to take into account volume. So here we go. So here we're not going to see an overall movement of water. We see also our vacuole is a little bit fuller, so it's got a little bit more water in it. But we will see just a general flow of water, both into and out of the cell, so in equal proportions. So it's not that all the water is leaving or all of it's coming in. Then on our far side, we're looking at our hypotonic environment. So in this case, we actually have a very, I switched colors. Okay, I'm going to keep with the same color if I can. A low concentration, so hypo. You might have heard this if you ever heard of someone being hypoglycemic. So hypoglycemia, it's like they have really low blood sugar. Okay, so this is hypo, so that means there's a low concentration of solutes in our environment. And there's a higher concentration of those solutes within our cell. Okay, in this case, we're going to see that water will be moving into our cell. And I tried to draw it so it looked a little bit engorged. So it's kind of like swollen. Now, there's a word for this, and that's turgid. Okay, or there's turgor pressure, which is associated with plants having plenty of water. And in fact, for plants, this is like a happy situation. They want to have water. On the other hand, this is not a good environment for plants. So this is called, plasmolysis, or this is when it's shriveled, okay? So this is when the plant cell shrivels, like we saw in our lab. So it's shriveled or it's plasmolysed. Okay. Then we'll look at our animal cells. So going down, we're looking at the animal cells, and we're trying to figure out how they're doing in each environment. So again, we're just drawing the particles. So we said we're going to have a cell. This is supposed to be my animal cell in my hypertonic environment. So that means lots of particles on the outside and very few on the inside. In this situation, again, we're expecting the same result. So we have water moving in the net direction out of the cell. It doesn't want to write there. Okay. So maybe I'll do another one just to show. Okay, water moving out of my cell. Right, it was hard to write there. And then on the other environment where it's isotonic, so isotonic, so about the same concentration of particles on the outside as the inside. So trying to get it as balanced as I can in my drawing. And remember here, we're still going to see some movement of water. It's going to be in both directions, so both into and out of the cell. So for an animal cell, this is actually our happy situation. Okay, this is where we're happy. Now this is not going to be good because the cell's drying out and shriveling, and that's bad for the cell. On the far end, we see that our cell is in a hypotonic environment. So in this case, there's a lot more dissolved particles within our animal cell than there are on the outside. In this situation, water is going to be flowing into our cell. So lots of water moving into the cell. And as you know, the animal cells don't have a cell wall, so do not have added protection. So this means that our cell is essentially going to explode or pop. Okay, so the cell membrane under a lot of pressure can explode or pop. And this is called lysis. Okay, so cell lysis is when the whole cell explodes. Okay, it pops. All right, so we're going to zoom out just a little bit. For some reason, the picture disappeared on my display, but I'm hoping you're still seeing everything that I would have seen. So, okay, well, I'm just going to keep going, hope for the best. So defining these conditions, we have the hypertonic. Remember, that's when there's a lot of solutes in the environment. Okay, I'll add in the environment, not within the cell. Isotonic, equal concentration of solutes in the cell and this time I'll add external environment just to be a little bit more specific. And that external environment. And then hypotonic says few solutes, okay, in the external environment. So then we're trying to go back to cystic fibrosis, and what the heck does this have to do with cystic fibrosis? So what we know about cystic fibrosis right now is that they have some trouble managing salt. So all I'm going to tell you for now is that cystic fibrosis patients are unable to remove salt particles from their cells, and it's not actually all cells, it's just some of their cells. So we're going to look at that more in class on Friday and next week. So when you're taking notes, I want you to draw out the pictures, okay, get all that information, and then this time at the bottom, as if you were doing Cornell notes, I want you to write a summary. So just a brief summary of the most important points, and try to write, let's say, I think you could write a little mini paragraph. So shoot for 4 to 5 sentences, most important points for these notes. Okay? Have a good day, and I will see you on, well, depending on which class it is, I will see you Friday, Monday. Okay?